Hi, Dr. McCaffrey. Could you tell us a little bit about yourself, like your experiments?
Yeah. So I am a postdoc right now at Stanford University in the lab of Mark Davis. I also did my PhD at Stanford in immunology, where I was in Mike Angelo's lab. And really, the major focus of my research is on tuberculosis, which is this disease caused by the pathogen Mycobacterium tuberculosis. And, despite being a very old disease, something that, has been afflicting humans for thousands of years, we still actually have very little understanding of its interactions with the immune system. We have very limited treatment options. Most treatments take minimum 3, but usually more like 6 months. And, it's one of these things where we don't have a good vaccine. There's the emergence of multi-drug resistance. And so it's a problem that I think is, very dire in a sense, but hasn't really been given a significant amount of research focus when you compare it to other infectious diseases like SARS-CoV-2 or influenza. And so, part of my motivation when thinking about what I wanted to research was I wanted to work on something that had a very relevant global health impact, something that was afflicting underserved communities that really had a clear need for additional knowledge. And then on top of that, I also became, from a more, I guess, conceptual point of view, very interested in sort of how bacteria interact with innate immune cells. And so these are the class of immune cells in your body that are sort of these first responders to infection. And, they oftentimes, when people talk about the immune system, they talk about T or B cells, which are things that can respond, for example, for vaccines and give you a memory response. But at the end of the day, the immune system is really regulated by these other cells, innate immune cells. And that really became my interest. I had studied microbiology in college at University of Maryland. And in the course of that, I realized I was much more interested in these interactions between pathogens in the immune system than I was in just studying the pathogens themselves and their sort of fundamental biology. And so that brought me to my, work now at Stanford, working on tuberculosis.
Oh, next question. I noticed that the paper you published in the Nature summarized the TB in human is characterized by the formation of granuloma in in affected tissue. The architecture and composition of which are thought to affect disease outcome. Could you briefly explain what this means?
Yeah. So TB is a pretty interesting disease or infection. I'll kind of use TB and MTB interchangeably. TB is technically the disease for the infection, but when most bacteria enter your body, and so in this case, TB usually enters through the lung. And so in a normal, say it was a bacterial pneumonia, what would usually happen is your immune cells would sense the presence of this bacteria. They would initiate this local inflammatory response where other immune cells come in to attempt to clear the infection. You might need to take antibiotics to kind of help. But usually this would lead to the immune system clearing out the infection and then you recover. Now, TB is a very tricky disease because the bacteria, Mycobacterium tuberculosis, is actually able to evade a lot of these early immune responses. And it actually uses immune cells called macrophages as its home, so to say. And so usually macrophages are these immune cells that will sort of eat or engulf bacteria. They'll digest them and then they'll sort of signal to the immune system that there's a foreign entity present. But TB is actually able to manipulate the macrophage and turn it into its long-term reservoir or house. And this really leads to this very dysfunctional immune response at that site of infection that ultimately leads to the formation of these structures called granulomas. And so you can really kind of think about granulomas as almost these balls of immune cells that have come to the site of infection to clear the TB, but they've gotten dysfunctional. And so instead of the immune system sort of clearing the infection and then resolving the inflammation, there's almost just this chronic inflammation where they're really just kind of in a standoff between the bacteria in the immune cells. And so, these granulomas, they can form during other infectious diseases and non-infectious diseases, but they really are one of these very characteristic symptoms of not just TB, but other mycobacterial infections. And so, kind of the idea behind this sort of relationship between the structure and the composition of the granuloma is that if you can imagine, if you have cell type A and you have cell type B, and those cell types need to interact to make a certain response happen, you would want them to be close to each other in that tissue. And what we actually see in granulomas is that there's this structure that form where a lot of these innate immune cells that you would want interacting with your adaptive immune system seem to be partitioned away from your adaptive immune cells. And so, this we hypothesize, and some other studies have shown, is really consequential because it really prevents the immune response from kind of kicking in to become very potent and clear infection. And so, this kind of fundamental concept behind my research was that if we could understand and sort of map not just the content of the granuloma, but the way in which those cells are organized and interacting, we might be able to better understand why the immune system is dysfunctional during TB. And in doing so, we might be able to sort of identify targets that could be treated with things called host-directed therapies, which are, instead of taking antibiotic to kill the bacteria, this would be a drug that you would take to sort of help your immune system or help prevent some of the dysfunctional either organization or states that we see the immune system form.
Wow. Mm. Is the architecture here means like the cell, they will change the shape?
The architecture can refer a bit to the shape of the cell. That's there's kind of different levels of architecture. There's at the individual cell level, and that actually is oftentimes an important indicator of what that cell is doing. In this particular study, the architecture here refers more to the way in which all of the individual cells are organized spatially. And so if you think of each cell as like a Lego block, and every granuloma is kind of an assembly of those Legos, the architecture here really refers to the way in which, those cells or those building blocks are assembled together. And the idea being that some granulomas are able to clear infection and they have their own sort of assembly or architecture, while other granulomas that aren't able to clear infection, have a very different sort of architecture or organization of those blocks. So it really refers more to the cell to cell interaction level.
Oh, like recently I read a paper related to the alveolar macrophages to migrate to the lung interstitium. So to disseminate MTB, is this also like architecture change?
Yeah, I'd say like architecture here is like really anything that captures sort of the way in which a cell is organized or positioned. And so the work you're discussing is really like an important first part of the TB response, where macrophages that I'm sure now live in the lung are often the first ones that will see the bacteria and engulf it. And then to initiate that response, they actually translocate to a different part of the lung. And that's supposed to allow for like the infiltration of other immune cells. And so, I think this could definitely sort of fall under the category of how a cell is positioned within the tissue itself.
Oh, okay. Next question. How does your research use biotechnology?
This is a good one because I think this was like really relevant to the work we did. So the kind of approach that I've taken in the labs I've been in have taken to studying infection and TB specifically kind of fall under this umbrella of what's called systems biology. And so a lot of, there's different ways to sort of take on a research question. And one way to take on a research question is to have a very specific hypothesis. Maybe you think cell type A or pathway B, I think, has this influence on this other, third thing. And then you can kind of set up experiments where you sort of assay that one particular cell type or that one particular pathway. Systems biology, by contrast, kind of takes this approach of saying, let's try and look at as many things as possible in as unbiased of a manner as possible. So we can really just get a sense of, what the sort of more multifactorial response is that's occurring in the tissue. And so when I first became interested in TB, one of the big problems is that compared to fields like cancer immunology or neurodegeneration, there really was a lack of studies that sort of took this approach to trying to profile many different parameters simultaneously. And this was something that ended up, I think has been a limiting factor in the field, is that most of the research will kind of exist on one cell type at a time. And you don't really get the ability to put all of these in the context of the entire picture. And so when I came to Stanford, my PhD advisor, Mike Angelo, in the Department of Pathology, he had actually developed a new method for imaging tissues that allows us to image, 40 plus proteins at the same time. And to give you a sense of that kind of advancement, most of the approaches before them would let you look at maybe, 2 to up to 5 in some of the more advanced technologies, proteins at a time. So to be able to go from 2 proteins to 40 proteins really allowed us to actually have the opportunity to take a tissue like a granuloma and map many different kinds of cell populations, many different kinds of functional pathways that those cell populations are sort of engaging for the immune response. And so I'd say certainly the biggest like technological application in this research has been the use of multiplex imaging. To give you sort of a sense of how that compares to some other approaches, oftentimes when you want to get a lot of information out of a tissue, you actually have to chop it up into single cells.
Mhm.
And then you can do things like sequencing or, cytometry. But if you do that, you kind of lose all this really precious information about how those cells were organized and interacting, because you can have all of the same cell populations, but if they're arranged in a different configuration, you might get a very different sort of composite response than you would in another sort of spatial orientation. And so certainly the multiplex imaging was the biggest one. In addition to that, there's a lot of sort of computational methods that go hand in hand with working with these data sets. Most of these are machine learning based. There's even now some approaches that are more deep learning based to kind of, identify significant patterns of sort of patterns that occur in the data. So the way in which cells are organized, we have methods to sort of detect those patterns and detect how frequent they are. And so that I feel like that those are probably the two sort of biggest areas of maybe newer biotechnology development that were relevant to this work.
Oh. The final question: what's your project's development potential?
Yeah, and so I think this study has kind of opened up a lot of future directions. So, at the time when we published this Nature Immunology study, this was really the first time that a high-dimensional imaging study had been conducted on human tuberculosis samples. And so a lot of the other work in the field had been done on model systems like mouse or macaque. And in doing so, we kind of identified some, I think, pretty compelling evidence that these innate immune cells are really driving the dysfunction of the immune response during TB. And that, oftentimes people have thought the reason that you have dysfunctional immunity in TB is because your lymphocyte responses, your T cells, are not doing a good job at responding. And this is true. But what we sort of postulated from this first study was that this is actually being driven directly by the way in which these innate immune cells are sort of shutting off the immune system. Like I think a lot of studies that sort of hypothesis or theory left us with a lot of follow-up questions about, if this is true, why are these cells doing this? How do they sort of get to this state of being? We're looking at granulomas sometimes months after the, TB disease has developed. So what sort of happened, prior to that to sort of lead to the current state that we see? And then I think really, all of this is with the, I think, big goal in mind of developing new therapeutic or vaccine strategies for TB. Because if we can start to sort of further dissect the different pathways, whether it be the metabolism of the cell, the different cytokine signaling pathways, we can sort of, almost like taking back the layers of the onion until we kind of hone in onto a set of features of that immune response that are things we can really target with these host-directed therapies. And, you know, it's always tricky because if you do something to influence the immune response, it's hard to just do it in the granuloma. So that's why a lot of drugs, you know, that are immune therapies can leave people immune suppressed. But our hope is that by doing a lot of these follow-on studies, both in human, but also some model systems, we can really hone in on sort of features that are very specific to the granuloma environment, and then try to develop therapeutics based on those.
Okay, thank you. I learned a lot about TB.
Yeah, if you're ever interested in, you know, please feel free to reach out. I think the more people interested in TB, the merrier. So, but yeah, thanks for the interview, and good luck with the rest of your course. And let me know if you need anything else.
Okay.